Hello everyone, this is Ravindra Babu. Welcome back to my channel Silver 90s Biounit. In this part of videos of molecular basis of inheritance, today we are going to talk about the differences between a prokaryotic transcription and a eukaryotic transcription, followed by post-transcriptional modifications. But before we going to start this today's topic, here a small request from my side. Please watch all my videos made on this molecular basis of inheritance so that you can understand the concepts clearly and completely. And one more request is please like and share the videos and subscribe the channel. Let's move on to the today's topic, that is differences between prokaryotic transcription and eukaryotic transcription. In prokaryotic transcription, the prokaryotic transcription requires a single type of RNA polymerase. This single type of RNA polymerase involves in the synthesis of all three types of RNA molecules. It can transcribe the m-RNA molecule and it can also transcribe the t-RNA, and this only can transcribe the rRNA molecules also. But the eukaryotic transcription requires three different types of RNA polymerases to transcribe three different types of RNA molecules. Those are RNA polymerase 1 is a type of RNA polymerase which transcribes rRNA molecules. It particularly transcribes rRNA molecules such as 5' srRNA, 18S rRNA and 28S rRNA molecules. Here you can remember this type of rRNA molecules transcribed by this RNA pol 1 by remembering the number 8 in all these three types of rRNA molecules. And the second type of RNA polymerase is RNA pol 2, which transcribes heterogeneous nuclear RNA. This heterogeneous nuclear RNA is the RNA molecule formed immediately after the transcription, and that consists of coding regions exons and non-coding regions introns. Hence the name heterogeneous nuclear RNA. This heterogeneous nuclear RNA, by undergoing a post-transcriptional modification, it becomes a matured functional m-RNA molecule. The conclusion is this RNA polymerase 2 transcribes m-RNA molecule. And third type of RNA polymerase is RNA pol 3. This RNA pol 3 is the RNA polymerase of eukaryotes which transcribes different types of RNA molecules. Here, besides transcribing a tRNA molecule, it also transcribes a 5S rRNA and a small nuclear RNA molecules. The next difference between these transcriptions of prokaryotes and eukaryotes is transcriptional factors. Transcriptional factors. Here, the prokaryotic transcription, it doesn't require additional transcription factors, but the eukaryotic transcription requires transcription factors during the elongation of transcription. And the next one is promoter sequences. The prokaryotic transcription requires two types of promoter sequences. Those are Tata box, this is otherwise called a Pribno box, which present after -10 sequence of nucleotides on prokaryotic transcriptional unit. And the next promoter is GC box, which is present after -35 sequence of nucleotides. But in the case of eukaryotic transcription, where it requires three types of promoter sequences, those are Tata box, which is otherwise called Pribno box, and there is another name for this eukaryotic promoter, that is Hogeness box. Hogeness box, which is present after -10 sequences of nucleotides on eukaryotic transcriptional unit. And the next type of promoter is GC box. Here this GC box present after minus 40 sequence of nucleotides, and the last one is CAT box, which is present after minus 110 sequence of nucleotides on the transcriptional unit of eukaryotes. And the fourth difference between this prokaryotic and eukaryotic transcriptions is transcription and translation processes in the case of prokaryotes occurs in the same compartment. Nothing but they occurs in the cytoplasm, because the prokaryotes doesn't consist of a distinct nucleus, thereby the process of transcription and translation occurs in the same compartment, cytoplasm. So that so many times translation begins much before this mRNA is fully transcribed. Therefore, the transcription and translation are coupled in the case of prokaryotes. Transcription and translation are coupled. But if we come to the eukaryotes, the transcription occurs in the nucleus and the translation process occurs in the cytoplasm. Nothing but these two events of central dogma of molecular biology are separated by space in the case of eukaryotes, but in the case of prokaryotes they both are coupled with each other. And the last and important difference of this prokaryotic and eukaryotic transcriptions are post-transcriptional modifications. In the case of prokaryotes, you cannot find any post-transcriptional modifications, because the prokaryotes genome that doesn't consist of split genes, no split genes in prokaryotes, hence no post-transcriptional modifications. What are these split genes? To understand these split genes, here we need to move on to the eukaryotic transcription. Say suppose for example this is the DNA or transcriptional unit of eukaryote genome, and which by undergoing transcription process synthesizes a primary RNA transcript. That primary RNA transcript consists of the coding regions exons, non-coding regions introns, like this. This coding regions exons codes for amino acids of a polypeptide chain, so that during translation process they involve in the synthesis of a portion of a polypeptide chain. But the non-coding regions introns, they do not code for any amino acids, so that they do not involve in translation, thereby no synthesis of a portion of a polypeptide chains. As these introns are interrupting or splitting the coding regions exons, hence the name split genes. The split genes is a characteristic feature of a eukaryotic genome. Here this primary RNA transcript, in order to involve in a translation, first it must become a functional matured mRNA molecule without introns. For this it undergoes a post-transcriptional modifications, during which this non-coding regions introns gets removed out. So this matured functional mRNA molecule consists only exons, which can only involve in the translation process and involves in the synthesis of a polypeptide chain. Here we can define the exons and introns in a different manner. Say this is exon. Exon is a portion of a DNA molecule which can undergo transcription and translation, right? Okay, and introns are the non-coding regions of the eukaryotic DNA which can only undergo transcription but not translation. They cannot involve in translation. Before the involvement of translation, the primary RNA transcript undergoes post-transcriptional modifications, during which these non-coding regions introns gets excised. The conclusion is this eukaryotic DNA and primary RNA transcript consists split genes. As they consist split genes, the eukaryotic transcription shows post-transcriptional modifications. But in the case of prokaryotes, no post-transcriptional modifications as they don't have any split genes. Suppose here the mRNA molecule formed immediately after the transcription in prokaryotes is called a nascent mRNA, which consists only the coding regions exons. There is no introns, nothing but there is no split gene, so that this nascent mRNA directly acts as a matured functional mRNA in the case of prokaryotic transcription. This is all about the differences between a prokaryotic transcription and a eukaryotic transcription. Except these differences, the transcription process in both prokaryotes and eukaryotes is almost same. Now let us move on to the detailed discussion on the post-transcriptional modification that occurs in eukaryotes after a transcription process. The post-transcriptional modification in eukaryotes followed by the transcription in eukaryotes includes 3 chemical modifications. These 3 chemical modifications occurs one after the other in a sequence. Those chemical modifications are, one is a capping, the second one is a tailing, and third one is splicing. Let us discuss one by one of these post-transcriptional modifications. The first one is capping. What happens during the capping step of post-transcriptional modifications? Say this is the primary RNA transcript formed immediately after the transcription in eukaryotes. This is otherwise called h-RNA or heterogeneous nuclear RNA, because it consists of the coding regions exons and non-coding regions introns, like this. This is the 5' end and this is the 3' end of this primary RNA transcript. During this capping procedure, an enzyme called guanosyl transferase. What is the name of the enzyme? Guanosyl transferase, which adds an unusual nucleotide to this 5' end of h-RNA, that is 7-methyl guanosin triphosphate to this 5' end of primary RNA transcript. This process of addition of this unusual nucleotide to this 5' end by this enzyme is called capping, and the added 7-methyl guanosin nucleotide triphosphate is called a cap. What is the function of this cap in this h-RNA molecule? This cap region protects this primary RNA transcript in the nucleus from the action of exonucleases, because the exonucleases, they cannot recognize this heterogeneous nuclear RNA as it consists of an unusual nucleotide at its 5' end. The another function of this cap region is to transport this m-RNA molecule to the site of protein synthesis. The sites of protein synthesis are ribosomes during the translation process. This is about the first step, that is capping. And the second event of this post-transcriptional modifications is tailing, during which an enzyme called polyadenylate polymerase, which adds a polyadenylate nucleotides at a 3' end of primary RNA transcript. This is what we call poly-A tail, added by an enzyme polyadenylate polymerase, which is a template independent reaction, nothing but for the addition of this adenylate nucleotides at a 3' end, this enzyme doesn't require any template. This is also a template independent reaction. Here also guanosyl transferase doesn't require any template for the addition of this unusual nucleotide 7-methyl guanosin triphosphate. What is the function of this polyadenylate tail? The function is it involves in the stabilization of this primary RNA transcript and it prevents this mRNA molecule to become double stranded. Say suppose for example this is a primary RNA transcript having AUC, a sequence of nucleotides, and this is GAU sequence of nucleotides. In the absence of this poly A tail, this primary RNA transcript may undergo folding like this, which results into a formation of base pairing between AUC triplet and a GAU triplet like this. Nothing but in the absence of polyadenylate tail, this primary RNA transcript may undergo folding due to formation of base pairing between complementary nucleotides, thereby formation of a double-stranded RNA molecule. If a poly A tail is present, it prevents this formation of double-stranded RNA molecules. That is the function of a polyadenylate tail. That is the second event of post-transcriptional modification. And the third event of post-transcriptional modifications is splicing. The splicing requires a special type of proteins called spliceosomes. These spliceosomes consist small nuclear proteins and small nuclear RNA molecules. The function of these spliceosomes is to excision of the non-coding regions introns and joining of the coding regions exons together to form a matured functional mRNA. So this is a matured functional mRNA molecule which consists of only exons. What happened here? These spliceosomes excised or removed the introns from this primary RNA transcript and made this primary RNA transcript into a matured functional mRNA molecule with a cap and a polyadenylate tail at a 3' end. Thus formed matured functional mRNA, it transported out from nucleus into cytoplasm to involve in the next event of central dogma of molecular biology, translation. The first two initial steps of this post-transcriptional modifications, those are capping and tailing, are very much helpful for the spliceosomes. Hence, the sequential steps of this post-transcriptional modifications are always capping followed by tailing, and capping and tailing followed by splicing. Here the reason behind this telling of this sequential order of post-transcriptional modifications is, in our textbook they have given in the diagram that capping followed by splicing, splicing followed by tailing, but that is wrong. This is the correct sequence of chemical modifications that occurs in post-transcriptional modifications. Keep remember this. This arrangement of split genes in eukaryotic DNA probably represents the ancient feature of genome. Nothing but in the process of evolution, DNA retained these non-coding regions introns, but RNA have got the capacity to lose these introns in the process of evolution by developing a special mechanism called splicing. Through developing a splicing mechanism in the process of evolution, RNA got the capacity to lose the introns. Hence, the presence of introns in eukaryotic DNA represents the reminiscent of antiquity, and the evolution of a splicing mechanism around RNA molecule represents the dominance of RNA world. This is all about a post-transcriptional modification. This is all for today's video. In the next video, I will come across the transcription unit and gene concept. Thanks for watching. Like and share the video and subscribe the channel. Vidyaya Amrutam Ashnite.